Protein synthesis is the process of creating proteins in a cell, and it occurs in 2 steps, and those steps are transcription and translation. It starts with transcription, which occurs in the nucleus, and it starts with a DNA strand we have here, and an RNA polymerase binds to the DNA strand, and it binds right before the gene that we're copying starts. So we're only copying like a part of a gene, and it binds to right before where that starts. And it moves along these bases and reads them one by one to make an mRNA, which is a messenger RNA, and that's just a copy of the DNA strand. Once that is done, the RNA polymerase detaches from the DNA, and the new mRNA strand is now free to leave the nucleus, and it travels to either the rough endoplasmic reticulum or a ribosomes in the cell. And it's important to note that transcription occurs in 3 parts, and that is initiation, which is finding the beginning of the gene. So that would be like the start sequence on our DNA strand. Elongation, which is making a copy of that gene. And termination, which is knowing when to stop, and that would be that stop sequence on the gene. And then it's also important to note the differences between DNA and RNA, and that's that it has a different code. So in DNA, the amino acid A would usually attach to T, so adenine to thymine, but in RNA, A would attach to U, which is uracil. So RNA does not have T. And then RNA is also a single strand, whereas DNA is a double strand. And then it is ribose versus a deoxyribose sugar. So DNA is that deoxyribose, and then RNA is just ribose. And then once that mRNA strand leaves the nucleus, it goes into translation, which occurs in the cytoplasm on a ribosome. And then it's important to note too that they read it in a 3-letter code, and that's known as a codon. So that will make sense in translation. So the mRNA strand and the ribosome, they bind together, where the ribosome can start building the protein 1 amino acid at a time. So the amino acids are brought to the ribosomes by tRNA, which is transfer RNA. And the tRNAs bring down the correct amino acids in the correct order, and they can join them together. So it matters that it happens in a 3-letter code. So if a tRNA would bring down the anticodon, GGU, then that would pair to the codon on the mRNA strand CCA, because that's what those amino acids pair together with. And then this process is repeated until there's a full chain of amino acids, and then once you have a full chain, they fold up on each other and create that final protein. And that is the process of protein synthesis.